Welcome to lecture 17 and 18. We will go over genetics. This will go over Mendelian as well as some more complicated genetics and Punnett squares. Gregor Mendel is considered to be the father of our modern day genetics. He spent most of his time breeding different pea plants and looking at how different characteristics were passed on from one generation to the next. He envisioned these as units of inheritance, and this is what we now know as genes. He determined various characters were heritable and that these characters could also vary amongst individuals. Some of these characters that he examined included flower height as well as color. He examined various traits and he determined that each trait was considered to be a variant of each character. For instance, purple versus white, tall versus short. He also determined that in true breeding plants have no variation for a particular trait. We now know that these would be considered either homozygous dominant or homozygous recessive. In Mendel's experiment, he carried out a series of hybridization experiments where he would take two pure breeding strains with contrasting traits and cross them together, for instance, purple and white flowered plants. When you're undergoing genetic experimentation, you can label your generations according to what you're crossing. Specifically, you start out with a parental generation. When you cross out those parentals, you get what is termed the F1 or first generation. It is important to note that if you want to carry on further to get an F2 generation, you would be crossing individuals from your F1 population. This is an important concept to understand, as some of the questions you will be dealing with will deal with parentals F1 and F2 generations. It's important to note that when you're dealing with an F2 generation, you're not going back to the parental to cross it together to get a secondary generation. In order to get your F2, you are taking individuals from your F1 population. In his experiment, we can clearly define these generations and what their characteristics were for what you were crossing. So the parental generation in his instance had the true breeding plants, purple flowered and white flowered. When he crossed these two together, he got the F1 generation, and the F1 generation were all considered to be hybrid because they had both traits from the parent. When the F2 generation was determined, he found some puzzling results. Once again, this F2 generation would have been obtained from crossing individuals from the F1 generation. Here we've got a depiction of those characteristics. So we have our parental generation, which consists of a purple flowered plant crossed with a white flowered plant. In the F1 generation, all of the plants were hybrid, but all of the plants had purple flowers. When these plants, the F1 generation, were either cross-pollinated or self-pollinated, the F2 generation that he obtained had both purple and white flowered plants. Remember, there was not much known about genetics and heredity at this point, so this was considered to be a very puzzling result, to get the reestablishment of the white flowered plants when you're only crossing purple flowers. This slide here depicts Mendel's experiments and the various characters that he examined, such as flower color, seed color, etc. Note that he took dominant individuals and crossed them with recessive individuals. Those would all have resulted in your heterozygous individuals. We can see in the fourth column that this gives the offspring ratio for the F2 generation. This would have resulted from taking those two heterozygous F1 individuals and crossing them with each other or self-pollinating them. We can also see the overall 3-to-1 ratio that resulted from those crosses, meaning that you got the reestablishment of the recessive phenotype when you're in your F2 generation. Mendel's work was pivotal in understanding that individuals can carry 2 traits for each character, and he also determined that these traits were determined by the parental generation. He also determined that for each character, one trait would be considered dominant to the other, so that even if you have both, only the dominant one is expressed while the masked trait is hidden. We term this recessive. So what does that mean in modern day language? We can translate that to understand that for every single gene that is present within an individual has 2 alleles for that gene, one from each parent. For humans, that would mean that we carry a gene, and one of those alleles that represent that gene would have originated from our mother, and the other would have originated from our father. If you have 2 of the same alleles, you would be considered homozygous or true breeding, and if you have 2 different alleles, you would be considered heterozygous or a hybrid. Mendel's work was also very important in determining the law of segregation. This states that during meiosis, for each gene, an individual's 2 alleles are segregated into different gametes. Recall that when we were discussing meiosis, we saw that the homologous chromosomes were separated out, such as the dad's original in copy would go into one set of gametes, and the mom's original in copy would go to the other. This illustrates the law of segregation. Remember that each gamete is considered to be haploid, so that means that it only carries 1 allele for each gene. This slide depicts that law of segregation. Note that we have our purple and white flowers in our parental generation. Keep in mind that when we're dealing with an organism is said to be diploid. As a result, the genotype or genetic makeup of the purple flowered true breeding parents would be big P, big P. The white flowered true breeding parent, because it has similar alleles for each of those traits, would be recessive and little p, little p. Now we have to consider what gametes are possible from that parent. Because that parent, the purple parent, only has 2 big P's, that means that the only gamete that it can produce is one with a dominant allele. Similarly, the white flowered, having 2 recessive alleles, the only gamete that it could produce is one with a little p. As a result, when you cross a true breeding dominant with a true breeding recessive, you end up with the F1 generation being hybrid, which is what we can see here in the diagram to the right. We see that we get all purple flowers because the pure purple allele is dominant. However, if we take a look at the genetic makeup of the F1 generation, they have both a dominant and recessive allele. So if we take a look at the gametes that individual can make, half of the gametes are going to have a big P, while the other half of the gametes are going to have a small p. Finally, when we determine what the F2 generation is, remember we are taking F1 generation individuals and either self-pollinating or crossing them with each other. So that means that in our Punnett square, we can see that in the sperm from the F1 plant, the sperm potential gametes would be a big P or a small p. And if we consider what the gametes would be produced from the F1 generation in the egg form, we would also have a big P and a small p. As a result, when you carry out the Punnett square, you are trying to predict what those offspring ratios would look like. As a result, in the first square, we have a big P from the sperm joining with a big P from the egg, which would lead to a big P, purple-flowered homozygous dominant individual. We would also have hybrids in two instances. Hybrids can be formed from either having a small p from the sperm and a big P from the egg, or vice versa, a small p from the egg and a big P from the sperm. So you can see that two of the squares in the Punnett square are hybrid individuals, and they have a big P and a small p, so they contain both the dominant and recessive alleles. And then finally, we have the fully homozygous recessive individual obtaining a small p from the sperm and a small p from the egg. On the left-hand side of this diagram, this depicts what is going on in gamete production. We can see at the top of this diagram, we are starting with a hybrid individual. This individual here carries two alleles for the trait. It carries a big A or dominant allele as well as a small a or recessive allele. Remember, when we undergo meiosis, we have a round of DNA synthesis or DNA replication. So as a result, each one of these alleles is going to be copied, and this is what we see in the second cell here. We are still considered to be in the diploid state because we have both dominant and recessive alleles within the cell. We can see that we've got an original and a copy, so we have two big A copies as well as two little a's. This is going to undergo the first round of division, meiosis 1. When anaphase 1 occurs, these alleles, these homologous chromosomes will be separated, and this is what we see moving from the diploid into the haploid state. At the end of anaphase 1, we would be considered haploid. This will then undergo meiosis 2 and will further separate out the chromatids, which is what we see in the bottom below. So as a result, after 2 rounds of division, we now have 4 gamete cells produced. Keep in mind that each one of them is haploid, so they each carry one version of the allele. We can also see that 50% of the gametes, or half of the gametes, contain the large A or dominant allele, whereas 50% of the gametes contain the recessive allele. This is our 50-50 genetic makeup for producing the gametes. This is what we can see in the middle of the right diagram, looking at the F1 generation and looking at the gametes produced. Half of them would have a big P, half would have a little p. This is equivalent to the big A and small a that we see in the left-hand side of the diagram.